Hello, this is chapter 5 Cell Structure and Organization, lecture number 2. The discussion topic for today is cell membrane. Cell membrane is the cover which is inner to the cell wall for the plant cell and outermost for the animal cell. Cell membrane is also called as a plasma membrane as it is going to cover the cytoplasmic part of the cell and differentiate the internal and the external environment of the cell. It is also called as a biomembrane as it is going to help for entry and exit of many solutes. Cell membrane is a quasi-fluid structure which is present both extracellularly and intracellularly. Extracellularly means it is present around the protoplast. Protoplast is nothing but the cytoplasm along with the organelles called as a protoplast. And intercellularly it is present around most of the cell organelles which are embedded in the cytoplasm in the eukaryotic cell. So basically we see this cell membrane is present externally as well as internally, helping for the separation of the cell organelles from the cytoplasm, which is also called as a cytosol. It shows the presence of lipids. Mostly these lipids are called as a phospholipids because the arrangement of these lipids is in a bilayer. So here we see that this phospholipid bilayer is having a phosphate head and a lipid tail. So here you can see this. There is a phosphate head orange color and a lipid tail. Both the side extracellularly as well as internally towards the cytoplasm. So here the phosphate head is hydrophilic in nature, called as a polar head, allowing the entry of water. Hydro is water and philic is loving. So it is a kind of a region which is having a affection for a water. Next comes the inner side of the cell membrane, that is the lipid layer. This lipid layer is hydrophobic in nature, called as a non-polar tails, and these tails are supposed to be hydrophobic, that means water hating, will not allow the water to pass through it. So basically we see that the bilipid layer called as a phospholipid bilayer are amphipathic in nature due to this hydrophilic and hydrophobic regions present in it. Next, this structure which we have just now gone through, it's a fluid mosaic model which is explained by or proposed by Singer and Nicholson in 1972. According to this model, the phospholipid bilayer and the proteins, we have seen that the proteins are floating. Some of them are embedded completely, spans through the cell membrane. Okay, so these are somewhat called as a peripheral protein, integral protein, depend upon the position of the protein. Apart from that, we see that there is a chain which is present on the upper side called as a glycoprotein, and the another chain which is glycolipid. So glycolipid means carbohydrate with lipids and glycoprotein means protein with a carbohydrate. So this is how the fluid mosaic model is making the protein channels which is helping for the entry and exit of the solutes, that is the molecules or the ions which are present outside the cell supposed to enter inside the cell, or maybe the one which are present inside the cell can go out of the cell. Next, so the structure is showing a pattern like a mosaic, and that is why we are calling it as a fluid mosaic model. Now comes the function. The function of the cell membrane is allow transport of the selectively molecules across it, and therefore it is called as a semi-permeable or selectively permeable. Two types of transport are taking place in it. One is the passive transport, which is simply passing of the substance from in and out, and another one is the active transport, which is supposed to help the solutes like ions to go across the membrane with the help of energy. So there is a basic difference between passive and active. Active transport is supposed to like flow of a solute from lower concentration to higher concentration. It is bit vice versa than what we are seeing in the passive. Passive is generally the flow of a solute that is from higher to lower. Water may also move by the process called as osmosis. We all are aware about osmosis. It is due to the uh, condition of a water, that means the solution which we are using, hypotonic, hypertonic, or else isotonic. There is nothing much to discuss in this because we already know that what the process of osmosis is. Then comes the polar molecule cannot pass through non-polar lipid bilayer, as I have already told you the prior slide. So they require a carrier protein, and hence those carrier proteins are nothing but the channels which are formed with the help of proteins which are embedded in the cell membrane. So this is all about the function of the cell membrane. Then comes the cytoplasm. Cytoplasm is the another important part of the cell which is supposed to be the grounded substance present inside the cell called as a cytoplasmic matrix. We also call it as a cytosol. It is a jelly-like structure called as a colloidal jelly and is non-static because it does not uh, try to remain at one place. It can move from one place to another so that it can help the cell for movement as well. For example, we all are aware of amoeba. Amoeba is having a pseudopodia which is formed due to the projection called as a pseudopodia. That pseudopodia is due to the cytoplasmic movement. Cytoplasmic movement means cytoplasm get aggregated towards the projection which is formed. We also call it as a cyclosis. Now the cytoplasm has got various type of organelles like endoplasmic reticulum, Golgi complex, mitochondria, plasmid, nucleus, microbodies, and cytoskeletal elements like microtubules. Cytoplasm also acts as a source of various metabolic activity which is taking place in the cell, and ultimately it is connected to the whole body. It also helps in exchange of material between various cell organelles which are present in the cell, and hence we see that there are some cell organelles which coordinately work together and form a endomembrane system of the cell. And there are also some organelles which are distinctly functioning, not included in the endomembrane system, like mitochondria, perform a specific function as they are called as a powerhouse of a body or a cell which produce energy ATP. Chloroplast is the another organelle which is supposed to carry out photosynthesis only in the plant cell. So here are some organelles which are distinctly performing their function. Next comes to endoplasmic reticulum. In short, it is called as a ER. Now this ER, if you observe, there is a diagram. The red color is the nucleus. ER starts from the nuclear envelope and ends up to the cytoplasmic membrane. So it's going to cover the whole width of that cell from nucleus up to the cytoplasmic membrane. If we see the network within the cytoplasm in the eukaryotic cell, except for the ovum and the red blood cell. So ER is present in eukaryotic cell. It appears like a network and it is forming a membranous tubules and a sac called as a cistern. Now here the total amount of a contribution given by the cell, 50% is contributed by the endoplasmic reticulum. This divides the cytoplasm into two parts. Endoplasmic reticulum is basically dividing cytoplasm into two parts. One within the lumen called as a laminal cytoplasm and other one is the non-laminar cytoplasm that is outside the endoplasmic reticulum. Basically endoplasmic reticulum is continuous as we have seen, and it's also going to help for the formation of a cell membrane. If you observe in the blue part, the endoplasmic reticulum has got orange color beads present on it. We can say that these are the ribosomes which are embedded on the endoplasmic reticulum. And the green color endoplasmic reticulum is having no ribosomes. This is a plain structure you can see. So these are called as smooth endoplasmic reticulum. So it's a clear difference. The endoplasmic reticulum having ribosomes are called as RER, rough endoplasmic reticulum, and the other one not having ribosomes called as SER, smooth endoplasmic reticulum. Now what is the function of this ER? ER is supposed to form or synthesis protein, and these proteins are further on wrapped and sent to the other organelle of the cell, maybe we can call Golgi apparatus. So ribosomes are also taking a part for the synthesis of protein. So rough endoplasmic reticulum is basically concerned or is working for synthesis of protein, and smooth endoplasmic reticulum is functioning for lipid synthesis. In fact, smooth endoplasmic reticulum also synthesize lipid, for example like steroids, okay, and it also going to act as a anti detoxifying agent. Detoxification is done with the help of the endoplasmic reticulum which are present in the liver cell. So this is how the smooth endoplasmic reticulum and rough endoplasmic reticulum helping for different functions of the cell. Next comes the Golgi complex. Now you can see the Golgi complex, the structure is given over here, and these Golgis are actually situated nearby to the endoplasmic reticulum, because ultimately we see that whatever protein is being manufactured by them, it's going to get packed over here. So we can call Golgi as a packaging factory. Okay, sometimes they also are manufacturing their own product, but they are also doing the packaging work, right? So if we see the structure of a Golgi, we see that there is a two phase given over here. The one is the cis phase and another is the trans phase. Okay, so cis phase is the incoming phase where the vesicles of the proteins which are coming from the endoplasmic reticulum will come and get fused over here. That means they will pour all their content into the lumen of this cis phase, and finally once that content whatever is brought by the vesicles is getting modified over here, right? So the modification is done little bit, and finally they are sent to the trans phase. There again they are packed into the vesicles and finally sent to the target wherever they are required. But the scene is, whatever vesicles which are carrying the content, maybe it is protein in the form of an enzyme, they will reach to the target because there is a stamping given on them. Something like some stamp is embedded on their vesicles so that they will reach to the particular site only wherever they are required. I hope this part is clear to you all, right? Next. So here I complete with this. Thank you very much. Chachi.